Okay, I think now we're good. Most of the participants are connected, so I believe we can start. Today we will be discussing skin aging with a special focus on models and assays for anti-aging research and testing. So this webinar is intended to last between 45 and 50 minutes and will be followed by a Q&A session at the end. Let's dive into today's topic. And for this, we will begin by the beginning. So why is it pertinent to study skin aging today? When we analyze life expectancy worldwide, what we see is that it has increased steadily over time, even if this tendency is more pronounced in some countries than in others, and it's expected to continue to grow. In other words, what we can say is that the global population is aging. Indeed, the World Health Organization estimates that by 2050, the world's population of people aged 60 years old and older will double, generating a marked demographic shift. And this, conjugated with the rise of beauty consciousness in society, explains why the global anti-aging products market is also expected to grow. But apart from anti-aging products, we would also add well-aging products. Anyway, you can see that this forecast predicts at least a duplication of the market value in the next 5 years. Here's one example, but still this trend is observed irrespective of the company performing the forecast. And logically, this information is of great relevance for the cosmetic and dermocosmetic industries. So now we will move to the definition of skin aging. But first, we should ask, what is aging? Well, in the famous review published by Lopez-Otin and colleagues, The Hallmarks of Aging, the authors describe the process of aging in general as a progressive loss of physiological integrity leading to impaired function and increased vulnerability to death. In this article, they describe 9 hallmarks of aging, 9 representative characteristics, and in a recent article from 2022, this number was extended to 14. As examples of hallmarks of aging, we can mention cellular senescence, stem cell exhaustion, mitochondrial dysfunction, etc. But what is important for us today in this webinar is to understand that skin aging is no exception, and that the traits that characterize aging in general also characterize skin aging. And what is funny to realize is that when we hear the term anti-aging, as in the title of this webinar, we tend to think directly about skin. And this is because aging of the human body becomes nowhere sooner and more immediately visible than in skin changes and hair graying, but today our focus will be on skin. In other words, the skin provides the first obvious signs of the passing of time, as Dr. Lehman put it in his book, The Remarkable Life of the Skin. Yet, it is important to keep in mind that the observable traits of skin aging are in fact consequences or the results of microscopic processes that take place in the body. In that sense, we can think of skin aging as an iceberg. So on the top of the iceberg, on the surface, we have the observable consequences, which are noticeable at a clinical level. This is that we can observe with the naked eye. Under the surface of the iceberg, we have processes that are not observable with the naked eye because they occur at microscopic levels. These processes can be studied at the level of tissue, cells, and molecules. Here is a non-exhaustive list mentioning some of the most important processes that we will discuss later in further detail, okay? One of the main messages we want to convey today is that understanding these biological mechanisms at the tisular, cellular, and molecular level is, of course, essential if we wish to develop new formulations or treatments that prevent or slow the skin aging process. So let's now see some factors that determine skin aging, which are several, in fact. Some we cannot control or modify. These are called intrinsic factors and are determined at the genetic level, but also by the passing of time. They determine what is known as chronological aging. Apart from the genetic background of the individual, we can also mention skin color and sex of the person, which are in fact ultimately determined by the genes. But again, it doesn't only refer to a predisposition, but also to the consequences of multiple cellular replications due to the passing of time. Then there are other factors that could, in theory, be modified. these are called extrinsic factors, such as sun exposure, pollution, climatic conditions, for instance, temperature and humidity, and environmental factors associated with lifestyle, proper or not enough sleep, tobacco consumption, nutrition, stress. These extrinsic and environmental factors are known as the exposome, a term you might already be familiar with. Briefly, the term exposome describes the totality of exposures to which an individual is subjected from conception to death. Let's now see some examples of the factors I mentioned. The first example, it is well known that intrinsic aging signs vary among ethnicities. So there can be differences in the clinical signs shown by the individuals of different ethnicities, wrinkles, pore size, etc., and also regarding the time of their life, the age at which the individual will display these signs. Here is an illustration taken from a scientific publication where we can observe real-life examples of these differences. Second example, sun exposure. This is the greatest contributor to skin aging. Some authors even say that probably more than all other factors combined.